All right, it is almost 9 o'clock in the morning, Eastern Standard Time. If you can't see the video, you should need to refresh your browser. Been on the seafloor for about 3 hours now. We are at the Damm hydrothermal vent field. It's located at along the Mid-Cayman Rise. We're at a depth of 2372 meters. This is an interesting site. It's about half the depth of where we've been diving at the Picard, about 20 kilometers away, up on a large outcrop called Mount Dent. And we are in an area with tube worms, those curly things you see on the seafloor, gastropods, shrimp, and squat lobsters. They look like crabs, but they're squat lobsters. And we are trying to position the vehicle so we can get and take a temperature of some shimmery flow at the base of that square rock right in front of the basket that you can see. Off to our right is a bag of cow bones that Cindy Van Dover deployed as an experiment. She'll be collecting those later this summer, looking at the types of invertebrate colonizers on organic rich substrate like cow bones or wood or whale bones, which is what you would normally find in the deep sea. So we're doing some maneuvering here, trying to see if we can fit the vehicle in between all the obstacles on the seafloor, mainly rocks. In our control van here, we have 4 different points of view of the seafloor. We're streaming the high def one to you. Cameras were acquired from James Cameron. These cameras were on the Deep Sea Challenger when Cameron traveled to the bottom of the Mariana Trench. And he's since donated his submarine to Hui, and we took the cameras and the lights and put them on Nereus to get a better picture that we could help us do our science and also send back to the world, courtesy of the live streaming ability of the Schmidt-Osten Institute's Falkor, the ship we're on. So this is an interesting area at the base of this huge hydrothermal mound. There's not a lot of vigorous flow here, but there's clearly a chemosynthetic ecosystem here with these tube worms forming the base of the food chain, the symbionts in their gut providing the organic carbon, and then other animals building off of that. So shrimp, gastropods, squat lobsters. We haven't seen an octopus here yet, have we? Sometimes can see octopus at hydrothermal vents feeding on some of these animals. Cindy says that octopus are often where there's bivalves, things like mussels and clams. And so far we haven't found any alive mussels here at Von Damm. We saw some dead mussel shells earlier in the dive.
We did collect one in 2012.
We collected one that was alive
Yeah.
in 2012. Part of our dive plan today is to search for more.
In fact, there's not much that needs two boats here. It's mostly just graze on bacteria.
It's mostly what?
Most of them will probably graze on bacteria rather than two boats. The two boats is in the two boats are the base of the food web. But there's not that much that needs to.
Okay, so we are setting up here at this rock at the Von Damm hydrothermal vent field, trying to get a good position so we can get a temperature.
So good morning again from the RV Falcor. The time is 9:05 here aboard ship on Eastern Standard Time. We've been having some joy the last couple of hours trying to get ourselves set up in this environment. There's one specific place that Julie really wanted to sample that had really good temperatures of fluid flow at this site. It was very hard to find them the first time we were here in 2012. When we did find them, it was such a neat place. We put out a time series experiment with those cow bones that Cindy Van Dover's been describing earlier in the dive. Unfortunately, when we came back to sample, we found out that the cow bones were actually fouling the underside of the vehicle and were making it impossible for us to actually get to the site to resample it. So we've, with Cindy's approval, we've just relocated the cowbone experiment, just moved it just a little way out of the way. And with that, some adjustments to the manipulator that Casey's managed to put in place, we have now remaneuvered ourselves and we are ready to pick up the temperature sensor for the super sampler. And we're going to probe that and see how hot those fluids are. There were a lot of places around here that only had 20 degrees Celsius fluid flow. When we were here in 2012, there was one particular sweet spot, which Julie thinks is this same place with the most vigorous fluid flow. We're hoping for temperatures here on the order of 50 degrees Celsius. And we're standing by now. Jill, do you have the ICL links working? Do you have the ICL working for the temperature? Because we're ready to start measuring temperature. Presumably once we get down there, Casey, it would be useful to change the view. So you can see the crack just above the black lid of the bio box. You can actually see some shimmering effect in the video. Casey's just getting ready to put the temperature probe from the super sampler into that flow. And we have Jill McDermott and Sean Silver at the back of the lab with their laptop connected. They're going to be ready to detect what the temperature is once we get in there. If we have temperature of about 50 degrees Celsius, then it's not just Julie who's interested in this fluid, but in fact Jeff and Jill and Sean will be interested to actually take their IGT, high temperature fluid flow samplers. They want to fire both those bottles in the same location. But we can't know whether that's the case or not until we've measured the temperature, so that's the first job. We just switched views now, so in our high definition cameras we're using this zoomed view. You can see some yellow frayed rope attached to instrumentation in the front of the basket, and just behind that, right next to some brown rocks with a white filament lining them on the right, you can see an area with fluid flow. It's shimmering in there. You can see shrimp down there. There are little gastropods in there. And now you can see the nozzle of the temperature sensor.
Alright, 6 degrees.
We have 6 degrees Celsius so far, and we're pushing further in.
10.
You can see a slight twitching in the arm today. Casey's concerned we have some water in the system. I see a little white rim around the top of the dark black rock.
5 degrees.
I can see, Casey, can you see there's a little white rim there? I wonder if that's coming out from underneath that. Is that getting it? 5.7, 5.5. 5.5. Feels like it's coming up from around the back of that rock, doesn't it?
5.3. 5.4. 7, 9, 12, 15,
Let's hold it there.
17, 19, 21, 22,
Alright, I gotta get checked.
24, 26,
This is looking good. We have
27, 25, 24,
So I'm going down a little bit, 23.
very difficult to hold position.
We're right around 23.
It's kind of jammed in there too.
Mm-hmm.
Is that okay?
Well, as long as the nozzle doesn't block, then it will. If it can flow, then
It's actually a little bit better that it is getting something.
23.
right. It's what keeping it stable, right?
Yeah.
24. Same thing, just landing and down. We're right around 21 right now.
Probably better.
We sampled 2 fluids here last year. One was 21, one was 50. Now we're at 24. 24, 23, 22, 21, 20, 21.
So it's a very tight balance here between as we're pushing here. If we push in hard enough, Casey can get temperatures over 20 degrees Celsius. If he doesn't push in hard enough, they drop away to 5 to 10 degrees. If he pushes in too hard, we start doing push-ups on the seafloor and pushing Nereus up.
20, 18, 17.
So it seems like this fluid here is all around 20 degrees Celsius, which is actually
That's gonna be a little cooler for a second.
slightly cooler than the surface ocean. No, we just
Here won't find a steady
So I'm
temperature.
not gonna kill myself because we know where it is.
so we know how to get to the good temperatures, Julie. We've just lifted out for a moment because we were going to push ourselves off.
Okay.
But as soon as you're ready to sample, we'll be able to put the nozzle back in and get that 20 to 25 degrees Celsius fluid.
How long do we want to do this for?
Half an hour.
Okay.
That was exactly the answer Casey wanted.
Are we long enough to know?
Can I just ask one thing, Casey? Can you see there's that rock with an angular crack down to the left? If you rotate it onto your left, what temperature would that be in there?
Stick it in there.
Do you see what I mean?
Yeah.
Just in that little crevice just there. Just we could, while we're waiting for Chip, we could just see if that's.
What is your next target? I'm not finding it. What's your next target?
There's the hot crack site. We don't know the temperature there. We could go check that out. And there's Twin Peaks. There's about 4 or 5 sites, including Old Man Tree in this way.
Okay.
We have Ravelin 1 and 2 that we can go to. And then we're going to work our way up. I mean, we could end up at White Castle and Ginger Castle. So all of those are on the possibility list for the day. What's the temperature just now? We just tried.
7.
Temperature right now is about 6.
Okay. We were just probing around to see if there was something better while we wait for Chip to show up.
So I'm gonna see if I can scooch back a little bit.
Okay, Casey's just going to reposition.
Okay.
Get a better setup. If we're going to be doing it for 30 minutes, we want to be as stable as we can be while we're doing the sampling. Do you need to change camera view before you move? Or are you good? So we don't lose sight of where we're going. So we are back on the wide angle view. And we're just going to make a pretty small adjustment, but a short adjustment is a long way in deep sea hydrothermal research. We found that the other day at the Pickard field. We could go 5 meters in the wrong direction and we had no view of the black smokers. In the same way here for this subtle diffuse flow that we're after. When we zoomed in, we only have a field of view of a few tens of centimeters at best. So we're just nudging ourselves back because the sampling that Julie wants to do will be a 30 to 45 minute iteration. So we need to make sure that we're not going to drive our pilots crazy if they're trying to do everything in a too delicate a position. So going to set ourselves up good and solid on the seafloor. Now Casey knows where we want to get the sample from. And then we'll get into that iteration as soon as we can. So we have the vehicle repositioned. We have Casey nudging the temperature probe and snorkel for the super sampler into just about the right place. We have Chip flicking through his notebook and getting ready to draw a sample. As soon as we get the okay from him, we'll get Sean to check temperatures as well. And when all is good to go, Julie will be recaffeinated, repastryized. There were some irresistible pastries at breakfast I heard everyone talking about. I snuck out myself just now. Had a couple of them. Little. It's almost like we had our own private boulangerie down in the galley today. Greg and Eric have a lot to answer for. Like why I wouldn't fit through the porthole in Alvin if I'm not careful. Okay, Chip is ready to start doing things. Sean, are you scanning temperatures?
I am scanning temperature.
Okay, Casey, let's stick the snorkel in. And we're looking for 20-degree fluids, so...
Well, we got up to 27.
So in excess of 20 is what we want, before we're going to know we're in the right place. So what do we have, Sean?
6.5 right now. Still 6.5. Dropping. 5.5.
I think left and down is probably the way to go. Maybe a little further left. See that brown rock you just touched in? And to the top of that looks more vigorous.
I think it's coming up from.
You think it's coming up from closer in? Okay.
And every time I move it, the float moves somewhere
Right.
else.
Where it's coming out from. We're on 5 or 6 now, Sean?
About 5.2.
Okay. It's going to dig in again.
6.6. 6.5. 8. 9. 11. 12. About 12.4. 13. 14.5. 15.2. 16.2. Still climbing quickly. 17.
Everybody hit the
18.
but
Still 18. 17. 6. Still 17. Dropping. 16.4. Climbing again. 17. 17.5.
Is it just in front of that then?
Passing 18. 18.4. 18.5. Increasing. 17. 17.8. 17.7. 18.2. 17.8. 16.9. 16.5.
Okay, we're just gonna try again.
Thank you.
so once I'm in, I can just turn the hydraulics off and the arms can't
Right.
move.
So you see where that shrimp is under that rock in front of us? Kind of right behind the plastic tubing just now. Let's try and I can see clear where the ruler is. That's clear there, and then the shimmering starts after that. So
Did it say grip lock on there, Mario? He's not. Oh, grip unlock.
6.4. 8, 9, 8.6,
come,
8.3,
you go down there.
9, 9.5, 9.5, 9.8, 10, 10.8, 11, 11.4, 11.7,
What happens if you go
12.5,
try
13,
try going left from there, maybe?
13.3, 12.9, 12.7, 12.3, 10, 10.6, 10.9, 13, 14. Where you just were, it was going up very quick. 11.8, 10.9, 10.7, 9, 8.4, 8.2,
And that's the same place that was 18 just now, right? Seems like.
11.9, 13.2, 14.7, 17.1, 18.1, 26,
Do it.
22.4, 20.0, 21.2,
We locked in there?
28,
Yep, we're locked in.
24, 26, 27.
Okay, thanks, Sean. We're locked in there. So we're starting purging now, Leighton.
T-max so far is 30 degrees Celsius.
Tmax of 30.
This will be new assembly.
That's correct. Chip, could you tell us what the port number will be for the sample? Super port. Thank you.
We're out of it.
Didn't move at all.
We're rock solid.
Just purging, right? Yeah, we're back at 10, 12, 14,
18, 20,
Okay.
21, 22,
We have the vehicle and the arm locked in place,
28,
so
26, 27.
So nothing is moved.
The
Right.
hydraulics are off, so it can't.
Okay.
we have everything.
Water wafting by.
We're
So
holding
it's-
right now at 29.9,
Right.
30.
So I think if Sean and Chip works together, when you draw the sample, we're in the right place. There's enough natural variability.
19,
Yeah.
18,
If you could tell us when you're ready to start sampling, please, Chip.
I will. I'm almost finished.
Thank you.
It's basically going between 12 and 30.
I'm purging. That's it. Stop now? You say stop.
watering it.
He's going to tell us when he does start. It's okay. Let's stand by.
Well, I'm purging.
But you just stopped and now it's coming back up.
Oh, so Chip's altering the flow.
Yeah.
You changing. It's drawing. So it's probably drawing faster through the snorkel than it's coming out of the ground.
Filter. That's why it's so hard to collect these types of fluids.
Yeah, that's exactly what's happening. Now we're back up at
Well, except that when it's going through the filter, the nice thing is it'll be drawing in, although it's in training, background seawater.
Slower if I filter it.
That'll probably keep the temperature up.
Can we run the pump slow when we're doing the bottle too?
I'm running it slow. I
Oh.
run even slower when I'm filtering.
Got it. Okay.
So when Chip's not pumping, we're holding it about 30 degrees.
So as you can see on the video, if you're watching online, we have the vehicle rock steady, we have the probe rock steady. When we turn Chip's sampler on and it draws water, the temperature apparently drops because it's actually sucking water in faster than it's coming out of the ground. So we know the fluid that we're looking at has a temperature around 30 degrees. Okay, we have purged the system. Sing out when you start sampling, please, Chip. Okay, started sampling. Very good. So the time is 13:29 UTC. Before you get too worried, everybody, wondering how you missed lunch, it's only 9:30 here on the ship. Breakfast seems like such a long time ago, and launch was even longer ago than that. It's just over 6 hours into my day, and we have found the first target and are drawing the sample. We're going to be taking whole water samples here. Doing a whole water sample. Then the second port. Okay. So the maximum temperature here is around 30 degrees Celsius. When we're sampling the whole water sample, which draws water quite fast, temperature drops to about 13 degrees Celsius. So you can imagine it's probably about 1/3 the diffuse flow, 2/3 ambient seawater, that's 4 degrees Celsius. We've just made the view a little darker while we're just conserving power on the batteries. Remember, Nereus is a battery-powered ROV. It comes down with a certain amount of power. We are currently at 63% of our total power for the day. So we are just conserving as much power as we can. We go through about, on average, around 10% per hour of our power. So we are in good shape. This was Julie's highest priority for the entire Von Damm site. I think that when she'd studied the microbiology at this location where the tube worms were, the populations were quite different from what she found elsewhere, both across the Von Damm site and at the Picard site. So if we can get this one sample today, we'd be pretty happy. It's the southernmost location we wouldn't have to come back to again. So we're going to take this sample for a few minutes. We're then going to move to a longer 30 minutes or so pumping for filtration, and then another water sample will come after that. And then we'll be able to move on to other things. And that'll be sample 04 for this dive. So, interestingly enough, to confirm our hypothesis, when we turned off the pump, the temperature came back up to 25 to 30 degrees. So when we pump more slowly for the next sample, which is going to be a filtered sample, going through a StereoX filter, we're hoping that the slower flow rate and that lower flow rate should then be available to then get higher temperature flow going through there. Jill, could I just see the time behind you, please? Thank you. So, time is 13:36. We have started sampling our second sample of this diffuse fluid flow. You'll see the picture is relatively dark on your screens at the moment. That's because we're conserving power. We've used about 50% of our power for this dive already, and we're trying to make sure we don't burn through too much. So since we're just sitting stationary, we're not using any of the thrusters on the vehicle right now. We're not using any hydraulics. Everything is locked in situ. Julie is perfectly happy that we sit as still as we possibly can while we're doing our sampling, and so that's what we're going to do.
So the temperature is about 25 with the filter and it's got a really narrow tubing.
And as predicted, Julie has just confirmed from the back of the room that on this sample, when we're filtering much more slowly, the flow rate coming out of the ground can be matched better to the flow rate going through the super pumping system. So we're sustaining 25 degrees C fluid temperatures, which means we're probably getting much closer to an undiluted sample right now.
Right, exactly. I'm not that, I mean, you know, we use the fluids for the activity experiments and things like that, so we're still getting that, but just being diluted, it's fine. I mean, I just don't think there's many other ways to get a fluid like this.
And we're predicting we're going to be doing this for the next 25-30 minutes. Very good. So 30 minutes will get us a large volume of fluid through the filter.
Yeah, we'll probably pump.
And that's the only thing that we're using for electricity right now. So we're just discussing plans of what we're going to do after we move on from this site. We think with the new manipulator capabilities we've got, that while we're set up where we are, we will put out our little marble planet that we have from Josh Simpson right in this location, in this field of view. It's like our square rock with the fluid flow has become a little icon for this end of the Von Damm field. And then we're going to head off to a new area which I think is probably only about 20 meters away, I think, to Hot Cracks. Mario's just going to get us a vector to that. So it's 34 meters away, and there's a series of 3 different sets of fluid flow all in close proximity when we get there. So that's somewhere with some higher temperature flow. It's somewhere we haven't collected fluids from before. So we're going to head to there. We'll probably fire our gas tight samplers there if the fluids are a suitable temperature, and hopefully do some more diffuse flow sampling. If we see an area with some sparse shrimp, we'll probably try and hoover up about 100 shrimp as we go. And we'll take it from there.
So for those of you just tuning in, we've turned down the lights to save battery power, but we're sampling diffuse flow here at the Von Damm hydrothermal vent field. It's about 27 6 degrees Celsius. We're at the very base of the Von Damm hydrothermal mound. All the way up at the top, there's 226 degree fluid. This is the only place that we see tube worms at the Mid-Cayman Rise so far. 2 species of tube worms are related to those found in the Gulf of Mexico and methane seeps. You can kind of see some of them in this field of view, but they're very small. They look a lot different than those the big Riftia or Tevia that you're probably used to seeing from National Geographic in the Pacific Ocean. And even though it doesn't look glamorous, we're actually sampling like 3 billion cells. 10 to the 9th is billion, right? Yeah. Yeah, so we're pumping about 3,000 milliliters of this fluid through a filter and collecting the microbial biomass. And in every single milliliter of that fluid, there's about 10,000 cells. So 10,000? 100,000. So if you multiply that by 3,000, you get to 3 billion, right? Isn't that impressive, Casey?
1 bajillion cells
3 billion cells.
per milliliter?
No, 3 billions and 3 liters.
3 liters.
Yeah. And inside those tube worms, if you were to dissect them and look inside their trophosome, there's like 3 billion cells, right? 30 million per gram of tissue. We did some calculation. Yes, everybody, trophosome. Trophosome? I'm probably using it incorrectly. Anyway, no one ever thinks about microbes unless they get sick, but most of the microbes on our planet are good for you and make your planet function. They do things like make oxygen. They do things like reduce sulfate, reduce hydrogen, oxidize iron, oxidize sulfur. The nitrogen cycle critically relies on bacteria and archaea. They can do denitrification. They can fix atmospheric nitrogen into a form that plants, for example, need to grow. Plants can't fix nitrogen. Our atmosphere is made up like 70% N2 gas, and only bacteria can fix that nitrogen into a usable form, ammonia or nitrate or other things. And inside your gut, I won't tell you how many bacteria there are because it might gross you out, but there's a lot. And it's why Casey can eat that pastry. And it will get processed efficiently because there's microbes breaking down carbohydrates and proteins and dairy and all sorts of things that we consume. So, yay microbes.
Yay, microbes!
Yay microbes! I'm singing the praises of microbes because right now we're filtering upwards of 3 billion cells.
Awesome.
Just if you were to count each cell as a sample, that'd be quite a sample list.
Your sample 3 billion.
Can you name them all?
24.
No, I can't.
Now,
I can't
Cindy
name them.
famously gives names to every shrimp she collects. She only collects 300 at a time, so I think Julie might run out of names.
So with 3 billion cells, unlike Cindy, who collects her shrimp and can dissect them and look at them under the microscope, a lot of microbes look exactly the same under the microscope, so we use DNA tools to tell the differences between them. We can't just categorize which species they are by looking at them, so we have to get molecular signatures that tell us their phylogenetic relationship. So we've been pumping now for 20 minutes? Why can't I see a clock? Yeah, but Jill's head's right in front of it. All right, we got five more minutes. Okay.
Maybe another 15.
5 more minutes before like another
Oh.
30 minutes. Is that what we're counting down to?
No.
Or it's
So,
just like,
no, actually, I did just. Yeah, so we're going to go another 15 minutes on the filter, and then we're going to go 5 minutes on a bottle, and then we'll be done at this site.
No, we won't. We'll be done.
What?
I have other things to do.
Not really.
Mario, don't touch anything.
Do you want to talk about biography for a while?
So, do I want to talk about biogeography? Biogeography of what?
Large animals that we've been seeing over here.
Well, I sort of already mentioned that these tube worms are related to ones we see in methane seeps in the Gulf of Mexico and Barbados, and that they're not at all like the tube worms we see at the East Pacific Rise or the North Pacific or anything like that.
Did you relate shrimp to the Middle Atlantic Ridge?
And the shrimp here are related to the shrimp that we see in the Mid-Atlantic Ridge and the Indian Ocean. And we see very different types of shrimp in the Western Pacific. So there's a very strong biogeography of vent fauna. And that was one of the driving reasons for coming here to the Caribbean, because we thought it's sort of a in-between point between the Atlantic and the Pacific. The Panama Canal used to be open, and there was likely some exchange of material between the Pacific and Atlantic, but it doesn't look like vent animals. It looks like they took different trajectories and colonization patterns. We still have some missing pieces of that biogeography puzzle up at the North Pole and the South Pole in terms of hydrothermal vents, and sort of every new system we discover helps put pieces of that biogeographic puzzle together. Biogeography of microbes is actually a lot more complicated because they're tiny little cells that probably have unlimited dispersal. But obviously a high temperature microbe that requires anaerobic conditions isn't going to be growing in the surface cold seawater of the Arctic. So what controls that distribution and how does that environmental selection take place is a really big theme in microbial ecology, especially in the oceans where you have currents basically moving everything around. And we've been trying to study that in deep sea hydrothermal vents as well. Our sample size has been pretty small, but we're building a nice database of information to try to make comparisons between vents from the Western Pacific, North Pacific, Caribbean, Mid-Atlantic, and Indian Ocean. So that's my biogeography spiel. And we're still pumping.
10 more minutes, no repetition. No deviation.
No surrender.
So it's just about 10 a.m. here in the Falkor. We've been on the seafloor for almost 4 hours. Feels like longer. In the van we have our chemists and microbiologists sampling low temperature flow. You can see around our sampling intake there's a bunch of shrimp. This is about 25 degree flow. And right now we're pumping this flow through a filter to collect microbial biomass. So those of you who are on Twitter, you can follow along at Schmidt Ocean Cruises or Jules Deep. You know, if you're in that really boring meeting and you can't see the video, but you still want to know what's going on in the seafloor, you can follow us along there. I was just pointing out that the filters we use are called Sterivex filters. They're really commonly used by microbiologists because they're a cartridge filter, which means they come sterile and they're easy to get a lot of fluid through. They're used in everything from water quality studies to this. So the deepest I've put a Sterivex is 5,000 meters, which was yesterday. I've also taken it to the base of Luihi Seamount in Hawaii, 5,000 meters. That was a Steripack, a little bit different. That's good product endorsement for Millipore. And I'm pretty sure when they invented them, they never thought that this would be a use of them. But it works pretty well. I did try to convince Millipore to give me free things, but I was not successful. I know, one of Matt's colleagues works there.
Every engineer I knew wanted to work at Millipore.
Oh, really?
Don't work at Cooley. Yeah, because Matt has a Millipore shirt.
Yeah, and we have some Millipore coffee mugs. It's an old Navy buddy, I think.
The Millipore connection.
The Millipore connection. Yeah, so this is a small cartridge filter, but they also make this big stacked filter to get lots and lots of volume through. So it's actually 4 filters stacked on top of one another. And we took that down to Luihi, and I was convinced it would collapse because it had there's so much potential for that volume to get an air bubble in it, but it didn't.
A little air won't hurt. A little air just goes.
Yeah.
A lot of air goes just on.
Yeah, your shrunken head from last night was awesome. Yeah. So last night we decorated some styrofoam cups and some styrofoam wig heads. We popped them on the CTD and we sent them to 5,000 meters. And they shrunk on down. They're super cute. And that's a pretty common souvenir from these deep sea cruises. They're fun to bring to your family and friends. And actually, there is video online. I'll try to find it and post it on Twitter, where a couple years ago, somebody actually filmed one of the heads shrinking as the ROV went down. And actually, most of the shrinking happens in the top few hundred meters. So basically, all that's happening is you're pushing all the air out between the spaces of the styrofoam. And you can never really predict exactly how small it'll shrink because it depends on the porosity of your styrofoam. So I know styrofoam's out of favor now because it's not a very green item, but it's a really good use for it. So don't stop making styrofoam cups, whoever makes them. We need them. Well, and those really flat, that really compressed styrofoam that you get, like at Dunkin' Donuts, those cups, those don't shrink. It has to be the big pieces of styrofoam. Yeah, exactly. That flakes off and gets everywhere in your house. You can still drink your... I guess I'm having too much product placement today. I'll stop. What? Yeah? Is there some other product I should endorse?
You may never have to write a proposal again.
What other products? Well...
Today, Julie is using her coffee mug.
It's a sharper image coffee mug. And I bought it at TJ Maxx. And I'm tweeting from my iPhone.
And remembering that YouTube is smart enough to recognize any music we play, and they will prompt us up for that.
Yeah, okay, I'll stop.
We can hear the algorithms churning now, trying to keep pace with exactly which reason Julie Google will be back in the waves.
We have 2 minutes left of this sample. Everybody's pretty excited about that we're almost done.
But there's more after this, right?
There's one more bottle, but that's just a few minutes. So one of the most interesting things about the Von Damm hydrothermal vent field is that it has really, really high hydrogen. It's up to 18 or 20 millimolar hydrogen and a lot of methane. I think 3 millimolar methane or something like that. And I mentioned the highest temperature we see is about 226 degrees Celsius. You can't get that much hydrogen and methane just from magmatic sources. And so we think there's some really interesting water rock reactions happening, something probably like serpentinization, which is when a olivine rock reacts with water and a lot of hydrogen is released in the process. And we believe this type of process has been going on pretty much since our planet formed. And some scientists believe that hydrogen and hydrothermal vents were the key to the origin of life on our planet. Because when you have a lot of hydrogen, you can make simple organic carbon compounds without you can make a sample at 1405. See all the brains behind Nereus.
Chemosynthetic shrimp all in one location. It's basically, as Julie was So we're just going to change our view. Actually, have you stowed the arm?
I haven't moved the arm.
Okay. What I was thinking is... No, it's okay. It'd be nice to get some nice video of the site as well before we put the marble in place. But we can do that anytime. So our audience is increasing rapidly. Now we've stopped doing the microbiological sampling. Julie did her best bet to make it attractive and entertaining for you, I know. But, you know, a bug is a bug is a bug. And Julie will be able to chastise me forever now when this gets recorded on YouTube. So Casey is just stowing the super sampler. And we're moving into our next iteration. That's not super important. We can work the cameras around that. Delicate touch from Master Casey, the pilot there. Just setting up the camera shoot. What we're going to do now, since it is quite an iconic field of view, you can see just north of the basket here, what you can see is the shimmering flow around these talc rocks that are the result of extensive alteration of ultramafic rocks. Julie was explaining earlier that that's what makes for the unique chemistry here. It's the unique geology that underpins it, that because of the unusual nature of ultra-slow spreading ridges like the Cayman Rise, that's not just what makes them deep, but it also means that long-lived faults can expose rocks that were actually formed deep within the Earth's interior, 6 kilometers below the seafloor, and have then been slid up towards the ocean surface along deep penetrating fault cracks. Those same fault cracks also allow fluid to flow and interact with those rocks, and that is what we think explains the very high hydrogen and methane and other complex organic molecules that we find in the vent fluids here. and then that then has a knock-on effect. And that's what we're trying to follow, is how that then influences the microbiology and the macrobiology of the same sites. And what we're just going to do before anything else now is we're just going to take the time to zoom in on this site. We can see tube worms in the field of view here. We see shrimp in the field of view, small gastropods, and the shimmering flow. If we can pan down a little bit more, please, Casey. That's good. Ah, I see. Let's go left. And then up again till we don't see the basket. I think that's probably where you, close to where you had it before. That's great. And let's just up a little bit so we don't have the bio box in the corner. Super. And we'll just sit here and collect about 30 seconds of video. If Jill could just duck her head, I can see the seconds. Okay, 14:20. And actually, is it worth playing around with the lighting? See how we might adjust the temperature of the image. It's not looking bad anyway. I think it's just the way we zoomed in. Oh, that's yeah. That's a bit too light, huh? Yep, that's nice. So just come up for 14:21, and we'll stay like this till it turns to 14:22.
Do it on deck. I'll pick one of them up on deck without it. Look what he's done. Picked up a dollar bill before. That's easy. Almost got a quarter before.
So we're about to move in about 10 seconds. Okay, Casey, that's great. Let's move to the next operation. Thank you. And what we're going to do next is Casey's going to pick up what we have in our basket is a globe, a ceramic globe. It's a pale blue sphere. When we put it down, we suspect it probably went in, unfortunately, in amongst the underside of the rocks. We had a nice idea of where we would put it up on top of the rock, but it seemed to roll down and lost it in a crevice. We'll give it a few minutes to see if we can see it. If not, we'll let the ocean claim its prize. That's right, as Leighton pointed out. I think Julie explained that she just took 3 billion cells from the seafloor. She plans to name them all the way that Cindy names her shrimp. And if we end up giving them one small planet back, that's a good trade. The nice thing about these marbles is because they're made of silica glass, they're basically made of the same material that the rocks are made of in the first place. So that it really is elemental recycling. When rocks get eroded away, the last thing, the most resilient thing you see, like sand on a beach, is made of silica. That's the same material that these marbles are made of. And I suspect because it's fairly rubbly nature of fluid flow, unless we're very lucky, as we dislodge one rock, it'll just allow the marble to sink further and further into the crevices. So I think for today, Josh Simpson, we'll say thank you very much. We have some beautiful photographs we'll share on the web tonight that Jill took before we deployed the planet. And what we will do is move on very soon. We're down to 57% of our power for today. So it's really time to get back on with our scientific program. Okay, so our next target is on your DVL nav screen, Casey. We are going to go to a site called Hot Cracks, which is north and west from here. There's a series of 2 or 3 sites of fluid flow. 2 of them are just, as it's described, cracks in the rocks, and then off to a little bit to the right, there's a place called Twin Peaks, which was 2 small chimneys when we were here about a year ago. As we head that way, if we see somewhere that might be suitable to sample shrimp, we'd be interested in that. And as well as the But apart from the shrimp, our next priority would be to find somewhere warm enough to be exciting to sample for vent fluids using the gas-tight samplers. I think we have a vector to that. It's about 30 meters on a bearing of 310 degrees. So we're just passing right over marker X18 as we set off. We can see more of these large tube worms ahead of us. And away we go, heading northwest, back along the spine of this ridge. So it's just gone 10:30 local time here aboard ship. We arrived on the bottom here just before, or just around 6:30, just before 6:30. So in the first 4 hours, I think we've probably gone about 25 meters. We're about to double the amount of ground we've covered in the dive. And as we go racing along, one of the things we did yesterday, if anybody missed it, was we went and recovered a fluid sample that had been sitting on the seafloor for about 4 years from when we last deployed it. At that point, Casey and Chris managed to actually set a new speed record for Nereus through the deep ocean. We clocked 30 meters a minute at one point. We're not going to be going quite that fast close above the seafloor, but what we're going to do now is, because this is an area we've explored previously, we can basically pick up and move from one location to the next, we hope, knowing exactly where the next point of interest is going to be. This area of traverse we first made with the little Hercules ROV aboard the Okeanos Explorer in 2011. At that point I was on the ship and Jill was part of the team who was following along on land. And then in 2012 we came back to this area again. We have abundant shells in front of us. I think again these are dead mussel shells. Let's have a good look and make sure we don't see any live ones. This isn't where Cindy thought she saw live ones previously. There are many places where we have seen dead mussel shells. It's been one of the frustrations of our 3 years of studying this area in detail. We keep seeing. Well, that's the question. It's one of the questions. We see them a lot downhill from sites of fluid flow, so we worry about whether or not they've been eroded, or whether they might be mussels that live perhaps within the sediment, and as the sediment gets eroded, they get exposed. It kind of speaks to the idea that maybe there's been another option is perhaps whether there's been more active fluid flow in the past, and then it's become relatively defunct and it just can't sustain the mussel communities, but the tube worms are making a living. When we first came here in 2011 with an ROV, Paul Tyler from the UK was with me and we saw one of the worm tubes, and he was very despondent. He thought maybe it was a dead tube and it was the last surviving remnant of tube worms ever to be seen in the Atlantic, and we'd arrived just too late. And then about a day later we found live ones, and then we found all these communities down here. So I suspect we might have just dropped off the top of the ridge again, Casey. This area does actually go, we get through, maybe if we use our sonar as well for targets, we get outcrops of the large rocks, and then we get sediment patches in between as we come up this ridge. There we are, it's looking lumpy again to our right. We should be very close to these sites now. See a Galatheid squat lobster, or whatever it is, a squat lobster there. And navigation should say we're pretty much right in the right place. I think we want to be a little bit more over to the right if at all possible. These sites were more over to the right side of the rocky ridge. More dead mussel shells ahead of us in our field of view. And from memory, the fluid flow sites were just where the rocks run out as you head back over onto, when we get back onto sediment. But we'll be able to tell them because they're bright white and they have shrimp clustered around them. You see it makes almost like a herringbone texture on the seafloor that when we first started spotting these mussels, they almost stained the same colour as the host rock. Something light just here. I see Galatheids here. There's more white material over to our right again. I think we may have just potentially come past it according to the navigation. It may be we need to cycle back round. That's kind of nice. We're looking southeast. We have the sediment there. On our west, there's some shrimp here, so we're getting close. I see a big mound. Oh, is that just sediment on the boulder there? And we have shrimp this way. Shrimp is a good sign. So we're looking in all our different cameras at the moment for different fields of view. And a temporary whiteout as we stir up some wash around us. Well, in Jason depth from last year it was 2372, so it may be a little bit shallower than that. But we're on a good heading here. Basically we're heading along that spine and we're on the west side. We have the sediment is just out of view to our left. We're not quite sure. It may be... What's that just to our... Can you see this? What we had were little crevices that actually had a higher concentration of shrimp. So that's just off to our left, I think, isn't it? This feature here, and then there's a white patch in the distance there. There it is. There we go. This little crack right here, Casey, right under us. That's one of our hot crack sites. You can see how it got its name. I wasn't being very imaginative that day. I just said exactly what I saw on the tin. Yeah. Let's at least get Sean, then we can do the temperature and the ICL link. And if it's the right temperature, we can get the fluid there. Yeah. So you don't want to set up that side. Can we drop a marker here, Mario, please? So we're dropping a marker here. We're not sure if this is hot cracks number 1 or hot cracks number 2. I'll have to go back and look at the video from last year latent to tell which one it is. But if we could enter in the log, it's definitely one of the hot crack sites.
So, I guess we should stick to the trend of the day and sample from above.
Well, you've been enjoying it so much so far, right? Why should we make your life any easier now?
Trying to perfect the art of setting up on somewhere zero.
There is a, well, if we've dropped a marker here, there is a second hot crack site and another one that had two little chimneys. They're all here and hereabouts, to within a few meters. I think one might be just either due laterally off to our left or ahead and to the left. So we could always scope out one of the other sites. Any one of the three is probably,
Chimneys might be hard.
yeah.
It's up to you.
I think I remember what that one was like.
So I think, why don't we've dropped a marker here, right? We could reproduce it if we just keep this in sight, if we just lateral to the left. Or if you want to pan with the camera to the left and have a look and just see what else is. I think probably Max would like to come back and slurp the samples out of here. See if we can do like a little hop to the left maybe. Yep. Let's do the time warp again. Or could you even come in face forward from that side? If you rotated round through 45 degrees to right, could you butt up against the base of that? And then maybe that's a better thing rather than to leave this target.
In my previous track record, there might be something actually further up that way.
Yeah, I think actually.
I bumped up against it. You're probably
So should we try and find the? Should we try and find the
The chimneys?
the chimneys?
Yeah, maybe hotter than here.
Yeah. So I think they're just like a little bit downhill to the left. So Casey, a quick update that for Jill's purposes, they're concerned that if the shrimp are living there, then it's probably only about 10 degrees and not hot enough. So if we carry on down to our left, which way are we? We're still heading southeast. So I think if we can lateral downhill to the left.
Anyone thought the Supreme Court was going to be so conservative? Like, upheld healthcare? I mean, shrimp are
Are these?
you're asking what temperature. You know, we used to kind of see shrimp like
We're just waiting. Yeah.
20 Right, but I mean,
Okay, so we know there are some other little chimneys near here. I am just going to go check the cruise report to see if I can get a good vector from hot cracks to that other site. James, I think he's only put one target in the underlay right here. So if we just stay put for 2 minutes, I'll be right back. So I think you see that white discoloration? That might be the next hot cracks, or it might be the chimney site. So for everyone watching at home, if you wondered why we went radio silent there, I actually went off and decided to actually refer back to work we'd done previously. And we found out that when we came here in 2012, when we were driving northwest, we went over a place called Twin Peaks, which is a set of chimneys, 2 little chimneys of hot fluid, and then came over 2 areas we named as Hot Cracks 1 and Hot Cracks 2. So we pretty solid. We're at one of the 2 hot crack sites. We're not sure which of the 2. So we're now on the reciprocal heading southeast, and it should be just a few seconds ahead. Let's have a look at this just here. I think that's a chimney. See this white thing? If we settle down on that, there's going to be hot fluid flow. Yeah, I can see it coming out the top. Yeah. Wow, navigation works, huh? Good job. We have found a chimney that Jill looks like she thinks she can do business with.
Is this gonna fall over if I hit it?
Yes. So let's get some video of it first while it's still there. So we'll just let things settle out there. Jill, I think we're interested enough in this place that it's probably worth time to go give Sean a prod. And then we'll come back and savor the moment. I do like how well our navigation works and our ability to find our way around. It does take... Once you've got your head around it, and sadly I have been a very sad individual. In the last 18 months I have spent hours of my life in my basement watching these videos over and over again. But we're in good shape now that we actually have something that Sean's first impression is also good. We have to be careful when we change the move the chimney plumbing so we don't cross the streams. But apart from that, we're going to end up singing the song, Who You Gonna Call? Jeff Seawald! The trouble is, it's much more fun when you're doing that on the midnight watch and you find a new vent fluid, because then it becomes time to get Jeff out of bed, and you can get the entire van of the Jason watch before I've had sing along to the tune of Ghostbusters. I'm like, hey, we got hot fluids. We got samplers ready to use. Who you gonna call? And yes, we do hope you're all singing along at home, even if you've never met Jeff. But if you sing real loud, children, he's fast asleep. He was up all night. He only went to bed at 5 o'clock. But if we all shout, so one more time, who you gonna call?
Jeff Seawald!
Yep, I think that's gonna work.
I ain't crazy, no offense.
That's a very foolish thing to say. We have some big hot ones we can park you in. The ROV is marginally more stable than the pilot at this time.
That's rolling into a slowly falling down the hill position.
What we're going to do is we can see we've turned off the thrusters. We're not moving around too much, but what we'd like to do is switch to our zoom camera when we get the opportunity and get some nice video so we can actually check out where the orifices are. And then once we've done that, we'll have a good idea of where to put the temperature probe and check the temperatures. Yeah, let's actually give this a sample number for some highlight video. Now, Casey, with your calibrated X-ray vision, can you predict what the temperature of those chimneys is going to be?
Hold on.
So we have a bet from Jill McDermott of 150 degrees. A 115.
Okay, Jill.
Sean Silver.
14.
114. Ah. Mario has worked out who has the best inside knowledge here.
132.
125.
Ah.
125, sure.
Sean had 125. I was going to go for 126. I'm going to go further. I'm going to go for 127.
127. Who else?
And Julie, you're not too late.
Julie, 122.
Starboard outer. Starboard outer.
I'm going to go for 139.
Ooh, you little monkey.
That's not as much better.
We've all declared our bets. Brian Midson, if you're listening at home, you can bet again. You were out by 100 degrees yesterday, but it doesn't mean you shouldn't continue to be optimistic on our behalf. Yeah, I mean, that is all mineral material that's being precipitated from, that's growing the chimneys. We are looking at a vent site and we're getting ready to sample it. So, have we finished playing with the lighting?
Yep.
Let's keep going for one more minute, please, Leighton.
Yep.
We'll just get one more minute of this video, since it is a very attractive little site. You can see these must be quite tiny little shrimp, I think, that are sitting around on top of the chimney. Now, the shrimp don't actually withstand temperatures more than 10 to 20 degrees Celsius, we don't think. Last year at the top of the spire, they didn't seem to live. They didn't like hanging out anywhere but I think it was like 11 or 14 degrees Celsius, something like that. We can be pretty sure that the fluids coming out of those chimneys are hotter, but through the walls, they are warmer than that.
Focusing on flow is difficult.
Yep. I don't think there's anything there that's focusable on. Maybe this shrimp down in the front left, and that little lump of rock there. By definition, the animals like living in the fluid flow, so getting the sharpest possible focus isn't really something that we do. But that looks pretty anyway. And the nice thing about video is you can actually see the motion of the fluids when we do this. So, Julie, out of interest, what temperature did you bet?
122.
So then if it goes higher, you're excited for us, and if you're lower, then you're delighted, because then you can get really good microbial samples. It's a bit like one of my favorite comedians in the UK used to do that. He would always bet on sporting matches, Peter Cook. He would bet against the team he wanted to win. Because the worst that could happen is his team won, and he got what he wanted. And if he lost, he'd win his bet. And that would be good commiseration. And with that little anecdote, we've used up the time we wanted for the video, so thank you, Casey. We are ready to pick up the temperature sensor and see Who's going to win the hot fluid sweepstakes today? The winner, the prize, is that Josh Simpson marble we deployed earlier. You can swim down and fetch it anytime you like. And if you think that's a bad bet, you should see what it was like when Casey had to drink that can of alcohol-free O'Doul's yesterday. He still hasn't forgiven us. So we're just changing our field of view now. And the next thing up will be a temperature measurement. So I think since this is a temperature measurement that we're not going to sample, we'll count the temperature measurement as a sample as well, please. Yeah. And Mario, if you have a moment, could we please drop a target here and call it Twin Peaks 2013? And we'll be able to compare later how well that fits with where we thought it should be. We think it must be pretty much straight head on because we went straight from hot cracks to here just the way we said we should, right? It was the right heading, the right distance, and the right chimney. Which I think means that one on the right is probably the second one.
Looks like it's been.
And it's got some fluid coming out of that. It just looks a bit more gnarly and rubbly. The concentration of shrimp here is maybe what draws the eye. Well, Leighton's just asked a really interesting question of how these rocks get used or how they relate to the fluid. And it is actually one of the active topics of debate right now that we've been working on, that Jill has been working with our colleague Frida Klein, who is currently field tripping in France. I doubt very much whether he's joining in right now. But there are basically two interpretations of all this rocky material that we see. One is that it was the original ultramafic host rock from the mantle, and that the fluid flowing through it has altered it so thoroughly that it's almost unrecognizable. So it's almost entirely made up of talc is the main mineral present. The alternative, however, is from what Jill has seen or knows from the fluid compositions, is that the same fluids could actually be precipitating this mineral directly as it mixes with seawater. So that it could be that everything we've seen today and in the entire cone of the Von Damm Mound is all chemical precipitate. And if that's really true and it's all precipitated from fluid like this, it makes you wonder either how long the fluid flow has been continuing at current rates of activity, or alternatively what it was like when it was really active, if this is a waning phase, if it's been much more vigorous in the past to grow these huge deposits. But I think the from the fluid chemistry, you couldn't argue that unambiguously right now, unless Jill has some clever tricks up her sleeve. Or the alternate approach is that Frida spends a lot of time actually taking these individual rocks and looking at the textures in them and seeing whether he can actually see. And one of the things you can do sometimes is, although the bulk composition of a rock's been changed completely, if you actually look at the rock either under a microscope or in thin section, you can sometimes see the original textures of what the original minerals were before they got hydrothermally altered, if it was indeed host country rock that has been chemically altered. And so that's something that Frida's actively pursuing. So we basically have two ways of going after this. It's getting further and further into the detailed geochemistry of the fluids, which is why we're sampling these right now, so Jill can continue that work. And also Frida's work when he's not having fun goofing off around the south of France like he is on vacation this week, can be actually looking at the rocks and coming at things from that perspective as well. So Casey's just doing a little bit of housekeeping, getting things tidied up in the basket. And now we're ready to go with a temperature probe measurement. Is that the arm down the bottom there? See a little yellow T handle sticking out from under there.
I'm just gonna pick it up by the ball.
Okay,
There's no way I'm gonna get it.
right.
So
Snagging. And there we go, ladies and gentlemen. Casey further perfecting his basket origami techniques. If you see the camera going out black and every now and then, that's actually not the camera screen, that's just us blindfolding Casey. Makes his life more difficult.
just like I'm fine.
Yeah, you got the T handle facing up now, right? So I'm just going to get Leighton to call up the log and remind us what the range of values is that we've been bet. So we have temperatures ranging from 114 from Mario to 139 from Leighton. Mario may have done a very shrewd bet there because Jill's the person with the most inside knowledge around here, we think. She has us at 115. Next one up is actually Sean with 125. So in fact, the two fluid specialists have pretty much the central zone staked out. Oh no, Julie managed to split them with 122. So I think somewhere between Jill and Julie is looking good.
I was close.
Casey's X-ray vision. I think I was most wildly out.
That's why I picked the hottest. Can we turn off the basket sun?
We're melting.
Alright.
We're doing really well actually, thanks Leighton. I was just checking out a few things. We are pretty close to we've jumped slightly ahead in the menu. We're actually at task 7 out of 10 for the day, and the only thing we missed out is task 6, which is to slurp shrimp, but we're going to be in good shape to do that right after we've done these things. We are ready. Now if we pan up, let's hope that chimney didn't just dry up. That would be rather spiteful of it after it hasn't moved anywhere for a year. So now we're just going to reposition ourselves. You can see the vehicle had just drifted off a little bit to the left. The chimney now is in the right side of your field of view. And we're heading back to get set up nice and close by so that we can sample the fluids from this site. You standing by with the IC links? Ready with the IC to tell us temperatures when we get there? Excellent. So what we're going to do now is we're using the inlet for the super sampler to check for temperature. We're doing that because that has a more forgiving snorkel. We don't want to risk putting our IGT samplers straight in this fluid till we know it's a good temperature, in case we get mineral precipitate clogging the snorkel and then the fluid won't actually get into the sampler. So we're using the super sampler temperature probe to check the temperature. As soon as we get a confirmation that this is a good hot chimney, then we will put that super sampler probe back and get the first of our 2 IGT bottles out to sample the fluids. We're probably then going to rotate into taking microbial samples from the same place, if I'm not getting too far ahead of the game. And then after that, what you can actually see living around the edges of this chimney are the sparse shrimp, the sparsely distributed shrimp. And they were both here and living in the hot cracks chimney. It's just behind us, just a few meters away. And that was something that Max in particular wanted to get samples of, so we'll be collecting samples here. We're probably going to be sampling in this area. This was a site that we didn't sample at all in 2012, but we did notice it was there in the post-dive studies we've done over the last year, checking out exactly where all the different sites of fluid flow are. We knew this was a place that would be smart to come back to, and so that's exactly what we've done today. And so the first ever sampling of this site begins just about now. Once again, we're just switching from distant wide field view to zoomed in focused flow, and that allows us to see exactly where the little holes are in this chimney structure and where the hottest fluids are coming from. So there's the lighting, there's the fluid, and we're about to start measuring. These things are fairly brittle, Casey, but they grow back, so you don't need to be too shy about putting the temperature probe in there.
22, 21, 18, 25, 32, 33, 34.
And we're still around the periphery.
46, 45.
I'm not sure we can see the interior of that one. If you see along the top, can you see a little white rim around the hole? That might be more one to go in vertically.
23.
Yeah, we're going to try a different orifice, Sean. There's one we can see the front of the orifice, so we're going to try and go in on the top. That's probably the best one, yeah. You may have noticed that the manipulator isn't staying quite as steady today as it did.
70.
Oh, we just dropped out.
60.
Okay. How about
4, 81, 86, 91, 94, 97, still climbing, 104, 111, 112, 114, 116, 118, 120.
Oh, Julie wants you to stop now. She wanted 122.
We're at 123.
Oh, Julie's out of the bedding. Sean, you've got it at 125 at the moment. But we just nudged out of the orifice.
So we're still climbing, so.
My 127's looking good here. Can you stick a little further in? There's no harm in putting it down inside there, right?
90, 101.
Shove it in so you can't see the hose clamp.
You want to see that little, there's a button to the right of that blue light there?
Yep.
Tell you where you push that button.
94, 99, 104, 107, 109, 104, 100, 103, 115, 116, 117, 120, 120.
This actually, you know what? This isn't the sample that we're going to take the sample with first, right? We're going to do the IGTs first.
We are.
So we don't really need to get Casey to put it.
No, we have.
We know enough now that we want to do an IGT sample here. Think about it.
Yep.
So we can stow that. Sorry, Casey, I was about to get you to do the wrong thing.
Roger that. Not doing
Great orifice
No.
for an IGT.
Hmm?
That's a great orifice.
Yes.
That's the orifice you want to
That's the one we want to stick it in.
One.
So, who are you going to call? Jeff Seawald. Da, 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 da. But you see those three orifices, Casey? What you have to be careful now is just make sure you don't cross the streams. Yeah, I think we already had some, but let's get some more. Yeah, can we play with the lighting? Or I think maybe you just did.
I just played with the stone.
Can we just pan up a little? Can we pan up a little?
Pan up a lot.
That looks good. Cool. What happens if we turn some out? Maybe the outer ones? Yeah. So 09 was the temperature. Do you have a time for the temperature? I think it's either 123 or 126. I'm not sure. We'll ask Corm when he comes back. That's fine. We started the video just now at... Thank you. So...
Hey everyone, come see how good this looks.
I think they probably are, yeah, and more of the shrimp. So, Sean, do you have a preference for which IGT we're going to use? Let's, if you don't mind, we'll keep steady for the next 30 seconds just to finish collecting the video, if that's okay, Casey? And then we'll start getting the IGT bottle out. And it's whichever one is furthest inboard is probably your favorite, right?
Always the one that's harder to get.
What was the highest temperature you saw just now? Do you recall?
23.
Curses. I'm not winning anything yet then. I might just go meet Jeff in the corridor now and tell him how I want him to adjust the calibration to make sure he gets the temperature I want with the IGT, because we're not quite sure. It could be that, or it could be different generations, or, oh, male, female. There you go. You're more of a biologist than me, Leighton Raleigh. I could tell you more about the rock if you like. That was exactly the first time, the other site we were at in 2011. I was busy getting excited about what the fluid flow was going to be like, because I knew we were scouting out sites where Jill could come for her thesis work. And I had voices from both the UK and the US screaming in my headset saying, "Look at that, there are shrimp, there are tube worms all in the same place." And there's a very sad thing saved for posterity online of me going, yeah, but the fluid chemistry is really cool. Being very, very churlish. Okay, let's end that as the video sequence. And let's start getting the IGT basket out, please. The IGT out of the basket.
We have a whole separate IGT basket.
If only.
Yes.
Yes.
So we're just reaching the manipulator arm as high as Casey can. We can stay on the seafloor till 4 o'clock. And we're about... We've still got something over 50% battery power. So we have, I think at rates of consumption, we're probably running like between 7 and 10% per hour of our battery. So we've probably got at least 2. So we can do the fluid sampling here, the microbial sampling here, and the shrimp sampling here. And then we should go head over to where the mussels are, from the photographs, and see if we can find mussels there. And then if we're still in the game at that point, we could go do some more microbial sampling or temperature probing. If you hear a rattling noise in the back of the background, we have some laboratory maintenance underway at the moment in the back of the control room. Lars the bosun and Lisa are working on that. Just making sure it doesn't slam so we don't get any inordinate noise on the video feed, audio feed. And the good news is the door still closes. So I think, Casey, so you know the iteration, we're going to do 2 IGT samples and then we'll be using that one again after that. And I think the preference will be the nearer of the 2.
Nearer of the two? You mean the closest to the camera?
Yeah. Which, according to my magic basket diagram, when we pick it up, will be revealed to be IGT number 7. And while we're preparing to do this, I noticed that Sean has taken himself off to lunch because he's not that fussed about bottle 7. His favourite is bottle number 8. I bet he'll be back before we sample with number 8. Jeff loves all his bottles, but Sean and I have a particular favourite, and it's definitely number 8. We have to get it out the basket first. Okay, here we go. We're ready to get the probe in the orifice and start working out what the real temperature is for this vent fluid. I'm still hanging in there for 127 degrees Celsius. Okay, Jeff, are you ready to call out temperatures to us? Excellent. So here we go, we're in the game. Everybody's paying attention. Door's not slamming. Are you ready to switch to the zoomed in camera again, Casey? So we're just switching cameras for the zoomed in flow. And there we go. Jeff could already see temperature anomalies even high up above the orifice. All the way in. That's looking good. Let's stop that. Oh, we just lost the grip on the bottle, Jeff. Oh, you can see that. So we're just going to use the it's taking advantage of the flat surface of the bio box. Casey's just very delicately put the sampler down so we can get a better grip around that T-handle. What we hadn't noticed there was we didn't have the full perfect grip around the system. The nature of that T-handle is designed so that you can actually get a grip where it can't spin around. So we're just going to come at this again. Yep. You back in the holster from there to get it better grip. So we're just going to reposition this back in the holster so we can go again with a better grip. While you're out of that, would you like a soda, Casey? Or something? You've been sitting in this pilot's chair for about 6 hours without a break at the minute.
Well, I took a break after.
Hmm?
I took a break for half an hour of fluid, yeah.
Okay.
I'm good, thank you.
Anything we can get you? Nope. Okay, good. So that was a good rescue job by Casey there. Gets us back in the game. In the meantime, we already know enough from the temperature sensing we did before to break Brian Midson's heart at NSF for the second day running. And Brian was worried. Actually, it's probably a good thing. He'll probably be happy on our behalf. He was concerned that that fluid may have been below 100 degrees Celsius. And we already know that it's over 120, so we're in good shape. And we have to remember, we have the advantage that we have ringers in the camp here with Sean and Jill, who actually made some of the measurements of last year at other vents that looked just like this, so we had a pretty good inclination of what we thought we were looking at. So if the temperature doesn't get hotter than 123 degrees, Leighton, who's going to win our bet?
The temperature does not exceed 120 degrees. So busy talking. I might not let you into it then. Okay, here we go. 123 degrees, while Julie's on 122.
And Sean's 125.
Chris,
So I guess
127.
So I guess it depends whether it was 123.5
Can't see, but it looks like you have.
I think I do.
from every angle we have. It looks like we're in good shape now. We've got a good firm grip.
Jeff's vote is 136.
If it's not too late. You know, but Jeff was asleep when we were doing the betting earlier, so he doesn't know what everyone else bet.
Yeah, but I told him what was going on.
Okay, so Sean's not going to let Jeff win. Sean's here to keep Jeff honest. And that in itself could be a full-time job. 127 if you're going to keep the chief scientist happy. But you have to cool it down to 55 to keep the program manager happy.
You already had 114, so 114.
Jill was 150.
I said one something.
Oh, no, Casey, you guys 132, sorry. Mario was 140.
So you got 9 more degrees, but you got to get it. You mustn't go too hot because Leighton's above you at 139 and I'm below you at 127. Julie's already distraught because it's too hot for microbial life.
What?
These temperatures are already too hot for microbial life as we know it, right?
As we know it. That doesn't mean I'm not going to take a sample.
Doesn't mean you're not going to find the hottest life we've ever found on the planet. Maybe.
32.
Don't get too excited. We're just putting the
That was just turning the lights on. Maybe not. Keep going left. Let's go left, yeah. You see the most violent flow is just behind the claw. One more degree, baby, 1 more degree. There's actually another nice intact orifice just over to the right in the field of view as well. Can you see that one, Chill? Downhill to the right. Let's see how this does. Yeah. Hey, Jeff, in that field of view, can you see on the right-hand side there's another little chimlet up over the peak and down to the right with a white rim? That might be more organized and less chance for seawater to get entrained. Of where we are now? You're going to come right there. And then down again, where he is right now. It's hard to see from here what's happening in 3 dimension over the rim of that, isn't it?
16, 61, 75, 87, 90, 88. 88.
Uh-oh.
What'd you get? Oh, I can see it.
Where?
It's there. You see with a little white lip around it, so you can see the back and the front of it.
Over here?
Yeah.
Below where the two?
Yeah. I think it's probably further in. You might need to rotate a little bit round to clockwise.
$53, 84, 112,
It's looking good. Just a little bit further away from us.
130,
You wait if you want to.
28, 29, 90. Minus is 130.
Yeah, you don't have to go any hotter, Casey. You already won the bet.
28, 31,
Can you hold it there?
131, 32. Oh,
What are we now?
110, 107, 122, 127, 130,
Good.
132, 134.
Undo it, undo it. The sampler is too heavy.
Yeah, I'm having trouble with the weight of the sampler and the arm's ability to hold it.
Don't turn off that going.
Okay, Jeff, how's that looking?
28, 29, 70, 50,
It's a li-
40,
He must be a little bit further away from us.
84, 89, 102, 105, 115, 120, 125, 20,
Oh, just jumped out again.
$60, 83, 112, 125, 113, 105, 76, 89, 95, 102, 110, 115, 120, 122, 127, 88, 124, 130, 73, 116, 120, 130, hold it right there.
We're trying.
136. Yes! 137. Don't move.
Okay,
138.
you ready to take this then, Jeff?
You feel stable?
Stable as we're going to get, I think, yeah?
Okay, here we go.
Let's get it while we can.
I lost. It's the new Mario.
1554. This is sample 11, bottle IGT7. Maximum temperature we've seen so far? What are we at? We're stable at 138. Are we happy in the back of the van? We're happy. Good. 138 degrees.
You starting now.
Already started. 1554.
So who is the winner?
Me.
Leighton
Did
by
you
1
go
degree.
over, though?
He was.
Oh, he's closest without going over, isn't it? Not,
Isn't it
not?
closest under? I think the pilot. I think the pilot makes the rules all the while the sampler's still in the chimney.
Closest without going
Oh, we're moving. Hang on, hang on, hang on. Are you still good temperatures, Jeff?
Yeah, 137.
Okay. The snorkel's just moving a little bit. Tell us as soon as you're ready, Jeff, for the sampler. We'll have it on the audio. The microphone was open the whole time. We'll get people to tweet Julie of what they heard Jeff say. We have 375,000 people listening in right now, according to our propaganda. Remembering that 98% of all statistics you hear on this channel are made up on the spot. Still sampling? So that is sample 11, IGT7 complete. So we'll stoker that and go straight for the second IGT bottle, please, Casey. If, yep. That's a very good question that we'll ask Julie when she comes back from lunch. It is one of those things, it's actually a really cute thing of why temperature-resistant microbes could be really useful if they had any medicinal applications. Because at the moment what we have to do is sterilize things before we put medicine in them. If you could actually have things that we could sterilize to 100 degrees Celsius, we knew that these microbes could survive. If they had medical things, then we could actually sterilize everything else, kill off all the known bugs. Because at the moment we heat things to 100, I think, is the typical way to sterilize things. Well, if they're good bugs. If the good bugs had medicinal properties to them, I mean, that's the nice thing. It's not just that there's wonderful new things that live here that we've never seen before, but there are potential applications of these things. It's kind of interesting how a lot of our curiosity-driven research quickly turns into things that are actually useful to society as well. We just didn't know it was going to be useful when we started it. $138 degrees. That's hotter than the White Chit Castle and Ginger Castle, isn't it? Or are they 150 something? Okay. That's hotter than. So that's hotter than Old Man Tree, right? So my due diligence in my basement paid off. And how was that? And was that? Did that have interesting stuff? It's actually kind of north of here, right? Revelin's still on this east side of this array. Yep. Because that's kind of that's what I've been thinking about from the geologic setting as I've looked at where these fluid things are, is whether they aren't all a linear alignment, which kind of fits to the Mike Cheadle theory of whether it's just a big crack propagating through the hillside, which would explain the entire linearity of this ridge if it could all be chemical precipitate but the flow's just become most focused right under the Von Damm. That's why that cone's grown up. But then the question is, are we going to have a whole bunch of those cones in the future from all these sites? So this is bottle 8. This is Sean's favourite. So we have good Chris, right? Yep.
Change over here.
So we're just getting ready for a quick changeover of pilots.
Enhance snorkels a little bit, so we're sampling this.
So it's midday here aboard ship. We've been on the seafloor about, let's see, about 5.5 hours. We have potentially another 4 hours to go. And we are going to be getting ready to take the second of our 2 fluid samples.
I always just hold the jaw.
Then once that's done, we're going to do microbial sampling. And once we've done the microbial sampling, we're going to sample some shrimp from this site as well. And then that'll see most of our objectives for the day completed. The last thing we'll then do is scoot up towards the base of the Von Damm Mound to an area where we saw our best photographs of mussel shells when we had Nereus flying over here in AUV mode, what must have been about 2 weeks ago now. On dive 53, we're now on dive 60, and we've had a port call into Jamaica and back out, and a complete changeover of the vehicle from one mode to the other in between times. And so what we're going to be doing now is going back to that area to see whether or not we can find live mussels. If we can, we'll scoop them up and put them in the bio box, and that will see us achieve just about all of our objectives for this dive. So Casey's just wrapping up his handover and debrief to Chris Taylor, and Chris is about to get back in the seat, and we'll get back to work. So Casey's off for a well-earned lunch, and Chris is ready to go.
You guys have been using the telephoto camera?
Yeah, we've been getting the snorkel over into the right kind of area because the zoomed-in field of view is so narrow. And then once we get close, then we switch cameras and go in. And Jeff's been calling out the temperature anomalies.
Not with this sample.
No, he didn't. He did try that just now, and then it didn't work. We started jumping back out. And I'm not sure if the snorkel might have got bent while he was getting it out, Chris. He might. The orientation compared to normal might be a bit strange. And we were using, when you do zoom in, there's a snorkel over on the right. The orifice on the right is the one that got least disturbed. The other one we had a bit of a jump and not, so it got a lot of seawater getting in.
Okay, yeah, we'll look at
The far right, so once you're ready to zoom in,
yeah.
we can steer you. And it's actually down and on the right hand side, but I can let me just use the indicator
Yep,
just there.
yep, yeah,
That little
yeah.
one there.
We're on the same
Great.
page.
Good. So Jeff, you ready to call out temperatures when we get close?
I'm ready.
Okay, and this is going to be sample 12, Leighton, which is IGT bottle 8.
Number 8. So it's centered up a little more. Then, that's really good.
Mm-hmm. I've got the Doppler position in the sample log as well, so we've got. And since we've got the bio box, we could see if we can pick up a piece of the chimney as well, maybe for Meg. It's pretty fragile stuff here from what I remember, but well, and from what Casey demonstrated as well. You ready to switch to the zoom camera? Okay, John, if we can switch to the zoomed in camera, please. Just take a while for that camera to come up. There we are. So now we can see the... So what can you see for temperatures, Jeff?
Yep.
It's probably warming up, right? We're looking for around 130, Chris.
Yep, absolutely. 16, 21, 78, 109, 71.
Just catching on the lip there, wasn't it? Looks good.
47, 73, 90, 103. Seven, 136.
It's just on the far lip of that little hole.
I need to reposition it.
Yeah.
That's fine. I can see it's what's
Can you just rotate from there? Just tip.
Well, according to the difference there, it's going to hold it. It's
Yeah,
got
just
a black notch there. Wedge it in there. 74, 116.
so really at the limits of what Nereus is capable of here. These bottles are pretty heavy for Nereus's craft arm, so at this full extension here, you can see the weight of the bottle makes the arm twitch up and down, something a real challenge for the pilots to anticipate what it's going to do. How's that, Jeff? Oh. Got a 116? Thank you. You logging as well? Are you logging? Are you logging as well? How's that, Jeff?
124, 120.
Bouncing back out all the time. What do you think about that bigger orifice down to the left now that wasn't there before? You don't think we could sit down deeper into it and get? We can at least see it now.
107, 130, right there. Can you hold that?
No. Bottle's already moved. How are we doing now? Oh.
I have had it in there several times, and as soon as I hold the arm, it just
Right. And the trouble is, Jeff's thing takes 2 minutes to draw the sample, and that's so.
It's not that I can't get in there, it's the mistake.
Right.
That's not for this. You can't secure this one. It's too
Seems like the arm can't actually hold the weight of the bottle, Jeff. Extend it out into that orifice. We could try going.
Rotation of the bottle or just the total weight?
I think it's
Sort of
bottle weight and angle,
on
right?
the side and then let you see.
Well, it's not.
It's not the wrist.
It's not. It's the twitching the arms do. Because basically, once I hold it, it just starts shaking.
We could try this bigger orifice that we couldn't. When you see in the center of the field of view, at the base, Jeff.
Yeah.
When we were getting weaker temperatures before, we couldn't actually see down into that. I mean, that would be the alternative. Would be ask Chris to try that one.
Sure.
Is that all right with you, Jill? I think the arms is getting worse with the water in the electronics. That this is why you always. I think the snorkel already got bent round the wrong way, so the bottle's in the right place. It's just the snorkel.
Try to see if I come in a different way. Maybe there's a
Try and use the weight of the bottle to hold it in place. What's the temperature there?
16.
Hmm. The hose clamp's right on the lip there, but the probe's right in the flow.
different spot you want to try.
What I was wondering is whether if you got right in down the bottom there, whether that's somewhere you could hold it more stably if it's a bit closer to you. And I think the danger is it's
117, hold it right there. Can you hold that?
Yes, it's frozen to the extent that it stays frozen.
if you want it, I would go now.
Need to drop to 84.
Came right out.
Okay.
Try the one to the left.
Yeah. Try that one down there.
Can you go to the hydraulic page and see what a hydraulic pressure is?
So we're just repositioning the sampler now. We're having trouble with our arm has been twitching around, so we're trying to get deeper into the chimney. Let's let that settle out and see what comes of it. Maybe come down to the left maybe when you get the chance. What do you see now, Jeff?
56, 45, 44, 51,
I think that black smoke shows you there's seawater getting into that.
65, 61, 58, 59.
Hey, Casey?
Yep.
Come here, I have a question. How come when I hit this index button, it says alarm master asleep?
I don't know. It's annoying.
It's like I never know if I have to look down every time.
You know what I've started doing? I hit it and then I wiggle it
What's
just
that?
a little bit to see. It doesn't take it, just hit it again.
Are you?
That's the way I've been doing it.
Okay.
I poked
So,
through the menus, but I
Chris,
couldn't figure out
the left side of that hole is where the hottest fluid is. Good, thank you. How's that?
122, 133, 136. Okay, hold it right there.
Okay, taking the sample. You taking it, Jeff?
Not yet. 130.
Okay, 133. We're taking the sample. Time is 16:24. (133) was max temperature. 138 again. Good job, Chris. No Julie sample comes next. Yep, and then the shrimp sample will also come from this site. The temperature holding up? It's over 100, right? Yeah. As you can see, our arm is continuing to bounce around, giving the pilots a huge challenge today compared to our past other days. I suspect there's going to be plenty of servicing to do tonight, but in the meantime, Chris is doing a masterful job hanging in there, actually inside the orifice. Fluid temperatures are staying up above 100 degrees the whole time, Chris. Where are we at just now, Jill? 120 something? How are we doing with the sampling, Jeff? Sampling still going?
Yeah, have about another
How's temperature now? Fluctuating.
Well, between about 110 and 130
Mm-hmm.
explosion right now.
So you can stow it.
Okay, all done. Good job, Chris. Thank you. Okay, Chip, you and Julie are going to be up next for some super sampling. So, yep, we're not going anywhere. We're going to be moving on to, it'll be sample number 13 from this dive. Nereus Dive 060 at the Von Damm hydrothermal field. We have just, yep, I was just going to say, we've just taken out 2 high temperature fluid samples for Jill's thesis work with Jeff back at Hui, and next we're going to rotate into doing some super sampling for microbiology, so I'm going to hand over now for Julie Huber is going to take over, sit in the hot seat, tell you nice stories about what we're doing, and I'll be back later in the afternoon.
All right. This is Julie. Back in the van. I guess it's not a van. It's a control room. Still feels like a van. Well, I know it's way nicer than the control van, but it still has that feeling about it. You're enclosed in a dark space with a bunch of monitors. So right now we're going to pick up the intake of the super sampler so that we can take some microbiology samples, the same site that Jeff and Jill just got their samples. These fluids are just a little bit hotter than the upper known temperature limit for life on Earth. The record holder currently is a methanogen that was cultured by Ken Takai, our Japanese colleague, who's out here on the Mid-Cayman Rise as well with his Shinkai submarine. And the way he got it to grow at 122 degrees Celsius is he put it under pressure to reflect the conditions that it actually grows at on the seafloor. So when we try to grow these organisms in the lab, we can do a lot of different things to try to replicate their environment on the seafloor. So in the case of a methanogen, we take away all the oxygen, we give them hydrogen, give them CO2, cook them at 122 degrees C, and add some pressure, and that's how we got that upper temperature limit for growth.
One question I had is, how do you sterilize this equipment? If these things can live in such extremes, how do you get it to a point where it's sterile before you actually sample it?
Sure, so a good question is how do we sterilize our equipment for microbiological sampling? So what we do before each dive is we basically flush the system with very, very clean water, and we fill all the lines with very clean water. So it's going down with basically the Millicu system water, microbial-free water. And actually organisms like methanogens hate oxygenated cold water. Methanogens from vents, they would probably die. So then when we turn on our pumps, we're sucking the vent fluid into the lines and then into a bottle or onto a filter. But we do have to worry about the transit time back, and I'm sure that many organisms don't like that long transit up into the water column, but we're still able to get them to grow and measure their activity back in the lab. It just takes a lot of finessing. What was that for?
That was for the
Oh.
big deal.
No big deal. So we're still trying to get our sampler out of the milk crate. Casey finally handed over the piloting reins to Chris Taylor. Yeah, I think is it caught on that bungee? That blue. Yeah. No, it's not. It just went free. So I think I've already mentioned before that Nereus doesn't have a lot of payload because it's a very light vehicle that's meant to go to the deepest parts of the world's oceans. So every pound on that vehicle is accounted for. Oh. So luckily we're in pretty flat terrain here. Hopefully we can just reach down and grab it.
Should be a bad potentiometer or sensor in there that senses its position.
Huh. Bless you. So we're trying to pick up this intake from the seafloor. Trying to gently lift up the length of tubing. So I mentioned that Nereus is a vehicle designed to go very deep, and so it tries to stay as light as possible. It's the reason it only has one arm, and even that arm was designed with weight in mind, as well as being able to operate under the pressure of 11,000 meters, which creates interesting engineering challenges for things like oil and hydraulics and stuff like that. So the basket is crammed tight with as much as we could fit on with our 110-pound payload limit that Casey gave us. Last I heard, we were at 104 pounds, so we're right on the edge there. What is that other cable? Is that the IGT or is it just for the temperature? All right, back in the basket and try to pick it up again. The flotation of Nereus is actually provided by these really cool ceramic balls, which, oh, is that a little jellyfish floating by? Whoa. It's a what? Oh, sea cucumber. Cool. Little holothurian just floated by. So those ceramic balls I mentioned, they look just like a, I don't know, like a baseball, except they're made of a really, really, really thin ceramic. They're hollow on the inside, and they're providing the flotation for this vehicle. And you can go to HUI's website to learn more about Nereus. I think there's a link to it on the Schmidt Ocean Institute webpage. And it shows some animation of the vehicle and also some of the different components that went into making a deep diving light vehicle. Nereus also has its own Wikipedia page. And in 2009, it traveled to the world's deepest part of the ocean, the Mariana Trench. Chris was there. And it was named for the Greek sea titan Nereus, who has a man's torso and a fishtail. It was a nationwide contest for high school students. And Nereus won out. So on May 31st, 2009, which happens to be my husband's birthday, they made it to the Mariana Trench. And the optic fiber tether is another thing that makes it really easy. Not easy, but makes it possible to go so deep. The optic fiber tether is about the diameter of human hair. It looks like fishing wire. And that's another engineering innovation that makes this vehicle possible. So question was if hydrothermal vents. Sorry, something just fell in the van. What was that? Oh. If hydrothermal vents occur in the Mariana Trench. So the trench is formed where the Pacific plate is subducting beneath the Filipino plate, I think. Jeff left. And when that plate melts deep in the crust, it creates a line of volcanic venting along the Mariana Arc. So they're called arc volcanoes. But actually at the bottom of the trench, you don't have a heat source, but you have subduction and recycling of rocks occurring.
But if it did occur, would it be incredibly hot because of the pressure field when it's up there?
Probably. There's some interesting mud volcanoes along the Mariana Arc as well, where pH 10 or 11 fluids are kind of seeking out from that subducting slab that's reacting with olivine-rich rocks. We've barely explored that part of the ocean. Nereus will be making a trip to the Western Pacific sometime next year, we hope, to explore vertebrate life in some of those deep ocean trenches. The Hadal zone, it's called, the deep, dark zone of the ocean where we know very little about the types of organisms that live there.
There is everything equipped with a fishery sound for when you
Nope, we've never put a fish sounder on it. It would just be payload, so I'm sure you could do it. So we're still working here to pick up our sampler that dropped off the front of the basket. Patience is a virtue in this field. Good things come to those who wait. So the way the arm's being controlled is using a, what is that thing called again? Master. You see a master controller. Maybe I'll try to take a, that's not going to take a good picture. Basically all the movements that the pilot makes up here with the arm are copied, are mirrored by the arm on the seafloor. The arm has 7 functions, from its shoulder to its elbow, wrist, and its claw. Its hand, so to speak. So keep practicing your video games if you want to be an ROV pilot. Tell your parents that's why they should let you play video games so much. Yep, but one of the challenging things about working with an ROV is that you're seeing a 3-dimensional world in 2 dimensions. And so that's why we have 4 different camera views to try to provide as many different points of view for the pilot and the scientist as possible. So we're only sending you 1 live feed, but we actually have 4 different views of the vehicle and the workspace and the field around us to help guide those sampling and exploring missions. Okay, we're going to be switching our stream over so that we can archive this 4-hour block. And you should tune back in about 2 or 3 minutes, refresh your browser, and we'll be here.